Hello, hello. I'm Madison, and I'm going to explain how to apply our pipeline, scTIGER, to your own research. I will also discuss a little about how scTIGER works and the types of conclusions you can draw from using it for your own analysis. This video does not explain how to install scTIGER, but there is another video that is linked on our GitHub page which explains how to install scTIGER. This is a brief schematic of scTIGER processing. I have provided a more detailed graphic at the end of this video if you would like to know more about how scTIGER processes data. scTIGER requires two input matrices, a case and a control. It offers several parameters to help you refine the output, and it also provides a GraphML file, which you can use to visualize the network on your own visualization software of choice. It further provides a histogram of the confidence of the edges, and a raw output file if you'd like to use the result in your own pipeline. First, we'll talk about the input. That's highlighted in the red box here. You heard me mention you'll need two files, a case and a control, but those two files can come from a variety of data types. There are two types of case and control files you can use. You can use data which is under different conditions, but of the same cell type. So that would be something like disease versus non-diseased, like prostate cancer versus normal prostate, as we have done, or an experimental dataset and a control dataset. The other option is that the data is under the same conditions and you look at different cell types or different cell lines. So you could do something like look at two cell lines with the same experimental manipulations, or look at different cell types in the same condition. We did endothelial cells and smooth muscle cells in prostate cancer, but obviously you could extend that to other studies. You can also use a variety of data types. scTIGER was only tested on scRNA-seq data, but it's reasonable to apply this to other data types with a similar structure. Some other data types could be single nucleus RNA sequencing, single cell DNA sequencing, and bulk RNA sequencing, but if you're using bulk RNA sequencing, you would need at least 20 different time points. However, those would all be potential applications for scTIGER. The only real stipulation is that the data must be in a CSV file and have the genes in the rows and the cells in the columns. And each gene cell coordinate must be a numeric value. But other than that, scTIGER can take any data type and perform this differential expression analysis. We have discussed the two required flags for scTIGER, but we also have a third required flag, which is the gene or genes of interest. These genes of interest come from prior knowledge about important genes for your study. When we were studying prostate cancer, we used AR because it's considered a master regulator for prostate cancer and prostate-related functions. So it's a good choice to seed scTIGER. But it is important to note you can change these genes every time you run it. So if you run scTIGER and it doesn't give you the output that you were expecting, or it's much smaller than you were expecting, you can go back and seed it with a different genes. You can also use scTIGER output to go back and adjust your genes of interest on the input side, right? If you want to see more regulatory patterns about a specific output pair that it gives you, you can put both of those genes back in and see how it builds that network then. We've discussed the required input parameters, so we can discuss the optional parameters. Each one of these optional flags or parameters has a default value, which means that scTIGER will run even if you don't specify a value for it. We allow you to change the number of permutations, which is the number of times that scTIGER will go through and use the deep learning program to infer edges. We allow you to change the top number of genes, which is the number of genes that are highly correlated to your gene of interest. If you make this value bigger, it will cause the runtime to increase. However, you will get a bigger picture of the whole network. We allow you to change the zero threshold, which is more of a normalization component, and that allows you to see if a particular cell has a measurement of 0 for 30% of its genes, it will automatically be removed from scTIGER. We've set it as a default to 30%, but you could raise or lower that depending on your study. We allow you to change the number of time steps between detected interactions. So 0 is the default because those are direct causal interactions, but you could extend it to 2 or 3 if you think that you want to look for additional genes of interest or you want to see how the whole network fits together. We allow you to change the starting point for scTIGER, though most of the time you will use the default 1. Default of 1 runs scTIGER and also provides you all of those GraphML files. If you change the start to 2, it will only generate the GraphML files from an existing scTIGER output. So if you want to change something like the alpha value and decrease or increase the number of gene pairs that come out of scTIGER as significant, you could do that by changing the alpha value and then changing the start value to 2 so it will run much quicker. We allow you to turn on CUDA usage. By default, it is set to off, so you would have to turn it on every single time you want to use it. You can change the name of the output directory with the -O flag. We by default have named it scTIGER output, but you could change that to anything you'd like. And the last optional flag is the alpha value, and that is the significance value for your negative binomial testing. By default, we've set it to 0.05, but you could raise or lower that depending on your study. Now that we've discussed the optional parameters and the input, you can finally get the output of scTIGER. And when you do, you will have a GraphML file and you'll build a visualization of that network. But what do those results actually mean and what can scTIGER tell you? scTIGER is responsible for creating a gene regulatory network, which means it tells you how genes are regulated by others. So you can look at it and say that gene A is upregulated or downregulated by gene B. You'll get many pairings and you can see how they all fit together. scTIGER is trained so that if you provide it multiple genes of interest, it will give you an overall global interaction map, as well as each individual one for your use. So now we've seen how we can apply scTIGER to our own data and the information you can discover from its use. If you'd like to hear more about how scTIGER processes data, you stick around with me for just a couple more minutes. Otherwise, I hope this video was informational, and if you have any questions about the usage of scTIGER, please reach out to us and we'll be happy to assist you. This is the full schematic of scTIGER usage. As we know, we start with 2 datasets shown in that yellow box up at the top left, both a case and a control, which have your cells as columns and genes as rows. scTIGER will take both of those and normalize them individually. Inside of this gray box, this is the part of the pipeline that is rerun based on the number of permutations you specified in that flag or that parameter. So both of those data sets will have random cells selected so that each has the same number of cells, and then they will be ordered on pseudo-timing individually. We use scanply's implementation of PAGA for pseudo-timing. Once that is complete, we will take the difference between the 2 matrices to create a differential expression matrix, which is denoted here as DEM. This matrix is subjected to Pearson correlation, and the gene pairs are then sorted based on how highly correlated they are to a gene of interest shown at the top right in the yellow box. Once we have our list of highly correlated genes to your gene of interest, we will go back to our differential expression matrix and only select those genes out of the matrix to feed to our deep learning program, TCDF. TCDF will go through and determine which of those interactions it finds to be causal. This then allows us to go back and determine the regulation type, if it is upregulating it or downregulating it. That entire process will be rerun for the number of permutations you specify, and then we will go and collect the results from all of those permutations. The results from all of the permutations will be collected and then subjected to negative binomial testing. The negative binomial testing determines a cutoff value for which gene interaction pairings are considered significant and which ones are more likely due to background noise. Once it's done that, it will generate the GraphML file for you and present that to you as its final output. If you have any further questions about scTIGER, feel free to email us or contact us via GitHub. We are happy to assist you.